I've had the privilege of being in the field of genetic engineering and recombinant DNA since it really began. Our laboratory in 1969 moved into this field, and we were able to develop the third enzyme used to split DNA. Fortunately, you get no prize for being third, but it was an opportunity to get into the field early. And I would like to do a few things with you. I'd like to describe my journey from an internship in pathology, where I was cutting people up and putting them into little bottles, and realizing for the first time the significance of life, and became absolutely unequivocally convinced that Jesus Christ rose from the dead and that he could be my Lord and Savior. And describe this walk through a time of working through, in the vortex of controversy, one of the areas that have influenced mankind. I'd also like to point out my usual disqualifier. Though still on active duty, all of the comments that I will make today are in my personal capacity, and I want you to know that I will be leaving the uniformed services in 11 days to become a minister in Fourth Presbyterian Church and adult education and adult ministries. Thank you so much. So as you did for Dr. Collins, I hope you'll pray for me as I move into Washingtonville in a different capacity than I've been during this time. I think it is appropriate to start right at the beginning, and I would like to call our attention to a verse that has been used before but must be repeated. In Genesis 1, in the beginning, God created the heavens and the earth, and now the earth was formless and empty, and darkness was over the surface of the deep, and the Spirit of God moved, hovering over the waters. And if I could have the lights down, and I believe the projector, I think, is on. And the perspective that I would like to bring to you at the beginning is looking at our sphere from outer space. Very few of us have ever left this sphere without dying. And one person was sent from God, both man and God, Jesus Christ, to come into this world to provide his word and our perspective on life. And in this perspective, as we looked at societal concerns for genetic engineering, these were the issues that were brought forward from Asilomar on, when we did a moratorium on genetic engineering, where we focused on the safety first, the effectiveness, the cost benefit, and then beginning the alteration of genes. And as I had the privilege of debating on platforms with Sinsheimer and Berg and others, whether it was appropriate, in my case, in view of mixing genes that were separate from the dawn of creation, or in Sinsheimer's view, from the dawn of evolution, or in Paul Berg's and some of the others, no problem, full speed ahead. And as we looked during that roughly year period of time, in 74, 75, we began to struggle with what should be done and how should it go forward. And as we looked at that, these were the perspectives that really began to emerge. That we would look towards gene products for therapeutic use, the diagnostic test, phenotypic gene therapy, and germ cell gene therapy. And in 1984, I had the privilege when Margaret Heckler, then Secretary of HHS, an extraordinarily devout Catholic, asked me to gave me the invitation to come into federal service as Commissioner of the Food and Drug Administration and bring online the research that I had been involved in genetics, in public policy, and in regulation. So as we looked forward on that, we first focused on the development through genetic engineering of medicinal compounds. And you've seen come forward a startling array of products that we never would have been able to have before. human insulin, growth factor, hematopoietin stimulating factors, a variety of medicines that were made possible. The diagnostic tests have also been brought forward in ways that we could have never anticipated at that time. We have worked forward on gene therapy now in an array of tests that I believe were as unknown to us and unimaginable. At present, and since 1987 when I made the ruling, the Food and Drug Administration has the responsibility for bringing forward all of the gene therapy products. Now, the first phase after animal testing is testing in individuals for safety and for effectiveness, and an IND is called an investigational new drug. Those applications are submitted to the Food and Drug Administration, and they're first tested for safety in about 20 to 100 patients, and from there, for larger safety, usually a couple of hundred, and beginning effectiveness, and then finally brought into larger clinical trials for effectiveness. And I show you this to give you an idea of the current pending, this was as of Friday of last week, the current pending studies at FDA. 142 studies are underway in gene therapy and 340 in somatic cells. An enormous explosion in this field. Now, what are our primary considerations for us as we look at this field? And my lament has been over the time that the Christian community has by and large been silent. There have been very little representations in the regulatory process or in the scientific process. And I'll show you some publications that can easily get accessed, and I hope that the Institute would have an opportunity to be a clearinghouse of information so that you might readily access this. But the dominant issue is creation versus evolution. And most of us, unfortunately, as professional physicians, scientists, Christians, live our lives in two compartments. What do we do on Sunday in our beliefs and where we are in our scientific thought. And I want to challenge you as strongly as I can to look at this issue. The next is, are we to exert dominion or are we passively involved in nature? Critical issue. Is there sin for which there was redemption by Jesus Christ, our Lord and Savior, or is this an accident? What are we here for? Is God in control, a passive observer, or in some individuals' minds, non-existent? And is man to glorify God or self? And that's a dominant question. And the thought patterns will vary as we look at this. For example, the thought patterns in creation are God created mankind and charged with the human race with dominion over creation. Creation, as defined, is making something from nothing. We as humans, in my opinion, are neither co-creators, unless some of you in the room have made something from absolutely nothing, but we are given dominion over, through God's word in Genesis, creation. We support through this a risk-benefit approach. We embrace the concept of no harm, and life has a purpose, and God is control of redemption, and revealed truth serves as our guiding principle. Now, in regards to the thought paradigm of evolution, and evolution is a very simple definition. It's the frequency of gene in a population as a function of time. No more, no less. But today it has a theology. And many of those tenets of the theology are listed here. And they are in conflict with each other. For example, life is a struggle. A variation of gene frequently leads to selected traits. And for those scientists in the audience who are human geneticists and not bacterial geneticists, I call your attention to an extremely important article in Science, Volume 272, it's June 1996, which is a punctuated analysis of evolution and its serial dilution of a culture bacterium over a 5-year period of time and glucose limitation, minimal selected traits that were of growth potential for, in millions of mutations over thousands and thousands of cell lives. Every one of us on this planet has mutations. Every one of us on this planet will be diseased. Leanne and I, my wife who's here with me and has supported me in all of these studies, have had 5 children, 2 of whom are disabled. We've lived with a variety of disease, and each one of us is totally biodegradable. We will leave this sphere in one way only. Now, another tenet of the evolutionary thought is there are unlimited resources that can be developed in and through technology. The little Jack Horner approach. Remember that story? Little Jack Horner sat in a corner eating his Christmas pie. He stuck in his thumb, pulled out a plum and said, "What a great boy am I." Notice the kid smashed the pie, dirtied his clothes, got the plum out and said, "How great I am." Problem with technology. Another thought, life is fragile, shouldn't be manipulated. Technology is responsible for social evils. Those of you that are in science, go back and read Sheldon Krimsky's book on the description of the emergence of the recombinant DNA. And a large part of the thoughts initially came out of the Vietnam era that science wasn't just. Science was not serving the people. That's where the strand, the largest strand of the objections came. Another portion is life is a lottery, and that one cannot manage it other than really randomness. A couple of other thoughts. Expert knowledge has managed the technology. The priestdom of science must be in control. You really don't have anything to say, people. Leave it to us. Excuse me, I slapped the mic. Leave it to us. It's probably a good punctuation. The other portion is it centered on man, or are all species equally subject to and have right for particular rights? And then ethics. The evolutionary scheme said we will devise the ethical framework. The creation scheme says the Lord has devised the ethical framework. And don't ever mix up the fact that there are two in those ways. Well, now as we look at this, let's turn to, with these 2 thoughts in mind, to the issue of gene therapy. Now, gene therapy, and I'll read this because it may not be visible in the back, a medical intervention based on the modification of genetic material of living cells. The cells may be modified ex vivo for subsequent administration or may be altered in vivo by gene therapy products given directly to the subject. The genetic manipulation may be intended to prevent the cure, diagnosis, or mitigation of disease or injuries to human beings. Where did it appear? That famous register, the Federal Register. Now, most of you will not read that, but the government requires all of the information to be placed here. And the slides that I'm showing you now are provided by the Food and Drug Administration by Amy Patterson, an excellent investigator reviewer who, as a physician scientist, has looked into and is studying this area of both science and regulation. Now, what is the challenge? The basic scientists and physicians remain skeptical of gene therapy because of the appearance that normal standards and scientific rigor have been suspended as we look eagerly to this. And we recommend that the applications of gene therapy should not be looked differently than all other aspects of medicine. It's very important. Though we're introducing genetic changes, as a physician, most of what I've been treating up to this time are phenotypic modifications. If any of you had insulin for diabetes, we are treating a defect either acquired or inborn. We're talking about here making a single correction genetically that might overcome that and its consequences, advantages, and disadvantages. Now, as we move into this, what are the types? We can introduce a gene in modified forms and in specialized cells. We can inject genes directly in the cells themselves. The gene can be put in through a gene vector. Vector can be produced and put into cells, or it could be expanded outside the body and transfused in. A wide array of techniques have become available. Back in the 70s, it was so limited that all we could modify was the cleavage of DNA by looking about from halfway here to the back door through a telescope at a spinning rod going in a solution of DNA. This whole array of dizzying technology has developed in little over 2 decades, an enormous change in science and society. Now, what are the kinds of things that can be done? An exogenous enzyme can be replaced, can be a natural enzyme or a recombinant enzyme. It can be done through gene transplants in cells to given to you as an adult or in utero. This is the array of technology that we're now looking at. It is built on careful diagnosis, the excellent presentations done on genetic counseling, the superb work that Dr. Collins talked of last night, and built as a public policy to look at and study and treat through genetic modification a number of diseases that have plagued mankind. Now, this is not too visible, but fundamentally, one takes a particular dysfunctional cell in this model that I'm going to use of manipulation, takes a diseased cell that one is going to work on, manipulates it, and this is a case of ADA deficiency. You add a variety of factors, and the right gene up there at the upper right, on your upper right, is introduced. The cells are forced to replicate, and then the cells transfuse back. So it is an introduction of a gene into a cell, the cell manipulated and transferred back. Now, what did we talk a lot about? The cartoon literature has captured our greatest fears. This was in early 70s when we were first putting in genes of frog into bacteria done by Stanley Cohn. Didn't turn into a prince, didn't go rebit, stayed E. coli, but our conventional literature picked that up. Ban recombinant DNA research. Counter demonstrations that would come up as well. All sorts of response on part of the public. This one was actually in 1970s, late 70s, when Jeremy Rifkin had some people come down singing to "We Shall Overcome, We Will Never Be Cloned," and unfurled at the National Academy of Sciences, "We will create the perfect race, A. Hitler, 1933. The concern of eugenics, the concern of gene modification. Profits. Bioethicists have profited from gene therapy, opened up whole new fields. So have regulators, whole new fields. Scientists. And as you can see here, gene splicing has been good to Professor Medrick. The whole concept of financial reward. This one I like because it helped the vegetarians out. If you can't read it in the back, thanks to genetic engineering, we now can all be called vegetarians, getting cows to grow on trees. This one I wish you could see. You can't, and I'm going to show it to you anyway, because this one, which appeared, has a combination in the containments, physical laboratory 1, 2, 3, and 4, of religious symbols. On that genetic tree is a helix as a snake, and offering to the scientist EcoR1, a restriction enzyme. Another woman is looking up there at other apples on the tree, full of religious experiments, and then at the back it shows escape and 2 E. coli mating in that cesspool that you cannot see. The mixture of religious symbols and totems with science. This is a slide that is so apropos to your excellent lecture, Elizabeth. What a beautiful baby. Was it produced by test tube cloning of the old-fashioned way? How are we going to look at these events? This I put here not with hubris, but it's consensus. A poor replica of me, of course. But FDA Young brought consensus to 23 countries. And that was when we were first looking at harmonizing the regulations over the world at OECD. Very controversial. How was it done? By consensus. Okay? Now, replacement therapy. A patient with a missing gene, in this case, peripheral blood or bone marrow taken, added the particular gene, amplification, bringing it back to the individual. One form of gene therapy. Now with this, I'd like to shift gears in order to now take from the 2 hypotheses of cloning with an evolutionary view, gene therapy with a creationist view, and walk you into an area that is going to be very interesting and important in regards to looking at this in utero with human stem cell therapy. I've selected this as one model to try to give those who are scientists in the audience a view of one particular nanotechnology that is likely to emerge that we can use as a discussion point. Now, what are the problems in dealing with inborn errors of metabolism? We are not focusing on a single gene, single enzyme defect in most human conditions. Remember, many of, and in fact most of the individuals that moved early into gene therapy came from a background of bacterial genetics. And they were used to looking at one gene, one enzyme. But it's going to be very hard to make that correlation. That the stem cells can be taken out and manipulated outside the cell, ex vivo, and then would have renewing populations, easily accessible, that can be put back into the body. The disadvantages are changes that may occur in that gene sequence that have been put in. You need to know that even though you put a gene sequence in, it can be edited, it can be modified, there can be post-translational effects, and you'd have to get it to go to the right spot. It has to home. Now, to give you an idea that this is not fantasy, this will show you the number of experiments that are going on and the rapid increase in looking at inborn errors of metabolism as seen at the Food and Drug Administration. Most of these are, and these are somatic cells, 79% deal with cancer, 11% with metabolic disease, 6% with AIDS, and 4% with others. So by and large, this type of gene therapy focuses on major changes that occur in particular individuals that have been already brought to this earth. I'm going to just show you a few of the many slides that I have on hematic cell transplant. And why is it done? The most important critical issue is that we are limited in postnatal bone marrow transplant. So that if we did the somatic cell transplantations on any of you, and I'll pick on Jay for the moment, if we were to do that on Jay, we would have to take out cells from the bone marrow to then take those out, amplify the gene, and put them back into a compatible individual. In stem cells, there is a much greater opportunity to do this technique. Here's the impetus. advances in the knowledge of how the fetus develops. And for those first 12 to 14 weeks, the fetus is immunologically immature, and stem cells can be injected. Advances in cell biology and advances in selection procedures. What are the goals? To establish normal cell function, to prevent the onset of a target organ damage, to achieve stable and durable chimerism, and to obviate the needs for post-birth therapies. Now, is this technology likely to explode on your scene, and will you need to be dealing with it? Absolutely yes. This is the number of projected bone marrow transplants with genetic modification as a function of time. It is really taking off at this moment. Now, the final slide that I'll show you on this is there's already been a number of cases, and that the relative immune incompetence of the fetus at 12 to 14 weeks enables cells to be introduced much more readily. Now, time does not permit to go further to demonstrate to you the fact that there are a lot of difficulties with this technology, as with all technologies. There's a variability of the genetic defect that one may be hoping to treat with gene therapy. There's a variability in chimerism, or multiple cells within the body. There's variability as to whether this will function. But there's enough promise that you'll see that. You will also see another topic that I would call for your ethical consideration, the use of other species cells, xenotransplant. And you've already seen that in the case of that individual with AIDS in San Francisco. But if these events are occurring, what is our role in the church? And let me spend some time on this. It was said very clearly, I believe, by Chuck Colson, that we need to do what we are called to do, to present the clear view of creation and of Jesus Christ as Lord and Savior, and to forcefully bring that forward within our professions. I felt that there have not been barriers to me in my life of bringing forward a Christian witness in a secular environment, whether it be academic or government. I think we really fully need to be present in the debate. Unfortunately, though our commander-in-chief sent his son into this world, gave us the guidelines, left and said, "Occupy till I return," most of us are missing in action. Full participation in the debates. That also requires an understanding of the technology and its interface with science and society. I would urge, as much as possible, that this organization be a clearinghouse. There's wonderful, wonderful opportunities for information. This is just one, 1996, Gene Therapy Resource Book, put out by the federal government. There's Federal Register summaries. A lot of information that can be brought on the World Wide Web, but most of us have not spent the time to be involved and engaged. We also need to be adding to this debate the absolutes. There are three ways in which decisions are made. Consensus, which essentially is a group of individuals sitting down together with the best available facts and coming up with a conclusion. That was essentially what we did for 24 nations in Europe. And essentially, we were focusing on those issues of safety, the issues of effectiveness, and trying to determine what the world should do in that regard. How should we go forward? Should we go forward? These that I showed you earlier. To deal with that, one has to be present and add your thoughts to that particular area, or my thoughts. The second type of decision-making is scientific, And I would emphasize that scientific decision-making is an approximation of truth. It is not absolute truth. It is an approximation of truth. The third type of decision-making is based on, I lost that, I don't need it again, Jay, thank you, it's all set, is based on revealed truth, absolute truth. Those three are the ways in which society works. And I do believe that as Christians, we need to add our perspective of absolute truth to this debate. What will this require? It will require the development of well-trained individuals that are multidisciplined, that can deal with both the ethical arena, the public policy arena, the scientific arena, and of course our faith, to be able to give a justification for the hope that lies within us. As we do this, I think there's a great opportunity to influence the debate, to be part of it, and I want to congratulate the organizers of this program for bringing together a stellar group of individuals to present, and for you who have come here to bring your wealth of information, because Leanne and I have enjoyed meeting so many of you and trying to see your perspectives, and we wish you Godspeed. I'd be happy to answer any questions I can.
Thank you very much. We have about 10 minutes for questions, 5 to 10 minutes, so if you'd like to move to the microphone, I know Dr. Young would be happy to address those.
Hello, I'm Janet Buckingham, and I'm from Canada, and I'm looking for some answers, and I've heard lots of good ideas, and I really appreciate your presentation, and I think that you might be able to help us with legislation we have pending. We have pending legislation before our federal government that would prevent animal-human crosses. You know, any kind of, you know, carrying of human fetuses by animals or any kind of genetic mixes there, and would also prevent any genetic manipulation or therapy which could be passed on to a future generation. Now, when I first read this before having come to this conference, I thought, that looks good. And now that I've heard from people who are much more knowledgeable than I am about these issues, I'm very questioning as to whether we want to prevent these things at this particular stage. And while I would like to maybe ask you on a one-to-one basis, what should we do about these specific things, my broader question is, how do we respond to these? How do we come up with the answers that we need to be telling government? Because when we're responding to specific legislation, we need to have very specific answers. Is this good or bad? Do we support it or don't we?
Thank you. Let me try to answer your question from a historical standpoint, and I'll avoid commenting till a one-on-one on your specific legislation. This really was not only a local problem in the United States, but a worldwide problem as we dealt with the whole field of recombinant DNA that was bursting on the scene. And there was a move in the United States in the 70s to bring legislation forward. Senator Kennedy was one of the leading advocates in that, and there were others as well in the House that brought forward bills. There was a great, and I think appropriate, focus on the fact that we should not have legislation, but instead should have guidelines. So the National Institutes of Health came forward with guidelines that were published. They had the effect of law, but they were not law, and they were modified over a period of time. Committees were established, and I was on the Recombinant DNA Advisory Committee, the first one that was formed, and many of the others that followed that, in which we had public hearings, public participation, published in the Federal Register the variety of decisions, and then changed as a function of time. I believe that that was the most helpful way to go forward. There were few countries in Europe that put absolute bans on. There were other countries around the world that had no regulations at all or no guidelines at all. In those that did have laws, they became very stifling, and they in fact were later subverted and eventually dropped. In the case of other countries which had no guidelines, it was Katie bar the door, and I think some inappropriate things were done. but we looked at the sanctity and dignity of life, we looked at controls on safety and effectiveness, and did it through guidelines. That allowed the plasticity for change. I think it was the right way to go. Every year, new advances will be made. Do we legislate against each of them or modify them that way? It's sort of like the Constitution. It becomes a living document. I think that way is a bit better. Jay.
Jay Holman, Baton Rouge, Louisiana, and Oxnard Clinic. Frank, the last time I tried to look at this very carefully, a year and a half ago or something, it was an easy decision for me. I just said, okay, somatic cells, that's just like sort of somatic cells, whether you introduce a virus into the genome or it's a temporary induction. That seemed quite reasonable, most like routine pharmacological therapy, that risk and benefit could be easily defined. And when it got to germ cell therapy, where you were introducing a gene very early in an embryo, using in vitro techniques, first it was very expensive type technology, so it wouldn't be widely applied likely, but the ethics of that always seemed to me, and many of the secular and and Christians at that point said, no, that's not for us. Of course, the Catholics rule it out completely because they don't want anything that's not natural in terms of reproduction. But for Protestants, I've had a patient who is one of the experts at the veterinary school at LSU who in these genetic introductions in animals, in transgenic animals. Of course, transgenic animals have been wonderfully helpful to us in understanding animal models, and I certainly wouldn't want to call a moratorium on that. But we're really trying to create transgenic humans by that, and the science of that, which I don't understand, although this fellow now tells me off the record, since I've not identified who he is, that he goes to South America, other places, and performs these same techniques on humans. And what is your feeling on the science of that at this point, and are germline or early embryo transfers something that Christians can advocate?
I believe, as I've looked at the in utero stem cell transfer, that though it produces a chimera, provides a great opportunity to prevent abortions of individuals with genetic problems that are severe, because one can potentially help that person that Elizabeth was talking about with grave concerns have a baby potentially where the defect of that specific gene, remember all of us carry genes with abnormalities, can be overcome. That really is on the borderline of germline, not quite there, but potentially. I do not see that as a significant ethical problem as long as all of the things that were brought into play so elegantly by Elizabeth are followed through on. In a single germ cell experiment, I'm still not completely decided, but a few more factors have emerged in my mind. Even though it's an introduction into the germ line, that germ line is not likely to be widely spread. We're looking at a fairly small number of changes. And our germ lines, from the time we're born until the time we reach our sexual maturity, are undergoing mutations that are random. And we don't count that. So this technology is not creation, it's manipulation of existing techniques and provides an opportunity to make a change in an individual. Now, I'm torn between the opportunity of doing it once and for all, if it could be done, with a person with diabetes once and for all making that change, or multiple injections of insulin throughout life. And as we go further down into this technology cautiously, through consensus, through the addition of some of the theological concerns, I think we'll be able to address a few of these issues. It's been properly, in my opinion, done very conservatively. Look at products first, diagnostics, the availability of somatic cell changes, and now in utero changes. Interestingly, the in utero debate is going to raise again the whole role of the fetus. Is the fetus a human being, and under what circumstances? And that whole debate of pre-birth and the rights of the unborn will be elevated on this. And in a sense, we're inching to the sanctity of the fetus through this action as well. But I only make that as a small footnote. So I'm cautious. I've gone and accepted in vitro stem cell, and now I'm struggling with germ cell, and I'm not there yet. Sir.
Dr. Young, I'm a recently retired general surgeon from Ellensburg, Washington, the other Washington. I was really pleased with the fact that you stressed creation today. I came from a science background, of course, medical school, and as a Christian had swallowed the theistic evolution line, and only recently I've come into the creation camp. I wonder, is there any other way that you can stress it, or stress that we take another look if we're in the evolution camp?
I really feel, as you have aptly said, that this is the most significant area that Christians have abandoned. After the Scopes monkey trial, we just were missing in action. And almost everything that I've seen in public policy focuses on those two worldviews. That's why I showed that first slide, looking back on the world. It's either an accident or it's created, and that changes everything. We're going to, in our adult education program, try to build a focus on about 10 of the great controversies that shatter individuals as they leave their Sunday school life and go into adulthood, and try to prepare both parents and young people for this. Evolution creation is our #1 topic. We're going to also look at good and evil. That's the fact that evil exists in the world today has been lost. Sin and redemption, some of these areas that we have not been able to focus on, and look at it from a scholarly standpoint. What are the facts and what does it mean? And not just from counting of days and of the models, because I used to worry what I would do in eternity. I mean, maybe you've never worried about that. But I used to wonder, what am I going to do? Would I be bored? And I realized that there's so much that I don't know about how God put this entire universe together. And I've got a little tutorial that I can sit in for millions and millions of years. So I'm not going to worry about the mechanics of this, because I'm not going to get that straight. But what it means in the thought process, what it means to have a creator that is Father and loves us. So I think we need to put the theories out, what it means intellectually, and expose the fact that evolution, instead of being the frequency of a gene as a function of time, has really gotten to be a theology of itself, and it has consequences. It's been a social philosophy, and a medical philosophy, a policy philosophy, and it undergirds much of what we're doing in genetic manipulation.
I want to thank Dr. Young very much, and appreciate the good questions. If you have further ones, you can catch him after. We're going to have a short break, just a couple minutes. Stand up where you are.